So we are really happy to welcome Dr. Patrick Worhunsky to speak with us this afternoon on creative uses of independent components analysis in brain imaging, both fMRI and PET. In fMRI, Patrick's been at the forefront of using brain networks to capture vulnerability in the addictions, especially cocaine addiction, as well as other disorders such as gambling that also feature impulsivity, compulsivity. These are research domains that are very strong interest to him. In PET, many of you are familiar with his work parsing the dopamine D3 and D2 receptors using the D3 preferring tracer PHNO. Patrick has an ongoing KO1 award, NIDA, that focuses on mGluR5 receptors in cocaine patients during early abstinence, and he has a host of productive collaborations in the past decade, especially with the teams at Yale, where he's currently an assistant professor in the Department of Psychiatry in the medical school. Dr. Worhunsky obtained his undergraduate degree at Trinity College in Hartford, followed by his D.Phil in the University of Oxford in UK, and then did two postdocs at Yale, one ending in 2015 in the psychiatry department, and the one after that in radiology and biomedical imaging. So he's truly a Renaissance man here, having his activities both in fMRI and PET. These are areas where sometimes people don't even speak to each other, but Patrick has these contained within one person. So we look very much forward to hearing today more from him about the powers of ICA. Welcome, Patrick.
Thanks for that intro, and thanks for the invite. I imagine most people have heard of independent component analysis, or ICA, before. Some of you may be using it, but I'd just like to start with a few minutes to just go over some of the concepts and basic principles of ICA, just sort of get us all on the same page, to have us better understand some of the data I'll be showing. And the goal of my talk isn't really to present to you some amazing discoveries about addiction that we've made with ICA, but more just sort of walk you through some data projects that we've had to give you a sense of the types of information that we can get with ICA, things that we can do with that information, and how we think it can be helpful in understanding addiction. And so the place to start is, what's the problem that we're hoping ICA can solve? And it's a very sophisticated technique to solve a very simple problem, and that's the problem of variance. And so variance is just, again, the basic idea of we run an experiment, we're taking multiple measurements, and those measurements are going to fall within a range of values and have a certain variance, right? And why we're interested in variance is if we think about something like abstinence in a treatment-seeking, substance-using population, it'd be really nice to know why some people are doing better during treatment, or perhaps why some people are doing worse in treatment than others. And we might be able to parse that apart by understanding what's causing this variance. And so variance can come in many dimensions, right? It can be between subjects, like this abstinence example. And we might have variance that happens across space, as it will in our PET data and the voxels of binding potential. But it can also happen across time, and we'll talk about that when we get to fMRI data. We're going to talk about sources of variance, right? What is causing this variance? Where is it coming from? And in some cases, that might just be true noise, right, or measurement errors or imprecision, and that that variance we can assume is going to be random. But on the other hand, we might think that we have underlying or non-random factors that are influencing the variance in our measurements. Right, so we can think of things that might be influencing how well someone does in treatment and come up with a list of things like readiness for change and social support structures that might contribute to whether someone will do better in treatment or not. Right, and those sources of variance are going to explain a certain amount of variance for each person. Right, and that brings us to components of variance. So components of variance are just how much variance is explained by each source. Right, it might not be the same for every person. But the idea being that these are independent sources of variance that are contributing to the variance, sorry to keep saying it, across our entire data set, and help explain in greater detail the measurements that we're making. Right, and so we can even just plot out, how much compliance is contributing to the variance of how well our subjects are doing in treatment. We're doing this quantitatively. Right, this is a computational procedure, and so we're getting loadings. We're getting source loadings, and we're getting component loadings. Right, and so source loadings are just how much of a component variance comes from each source. Right, and a component loading is just how much variance that source contributes to each measurement. I know it sounds circular, and that's because it kind of is, but it makes more sense as we get further along with the data. But these loadings are just betas or coefficients of variance in a linear mixture equation model. Right, so if we look at our readiness for change source, our source loading across our data set is contributing to, let's say this number represents 30% of the variance. And for subject 1, they've got about 25% of their variances explained by readiness for change. Right, we can multiply these things together and recapture how much variance is explained for that subject for change. And we can do the same for our other sources. If we multiply all of our sources for each subject together with their component loadings, add them up, we can recover the amount of days absence. Now don't pay attention to the actual numbers, I just made them up for example. But these sources are going to be different for, the component loadings are going to be different for different subjects, right? Our source loadings are going to stay the same, but this subject has more readiness for change, but far less treatment compliance and much less social support. And so they do far worse in treatment, right? So the idea is we're trying to reconstruct where all of our variance is coming from in our data set. And we're just going to be doing the same thing inside of a brain. Right? Our source loadings are going to be coming from voxels, or located in voxels, and our component loadings will just be explaining how much of each source brain is contributing to that subject's variance. I should say if there are any questions at any time, I can't see any hands or any other people, so just shout out if you have questions. But that also brings me to networks, right? We're going to have source networks and functional networks. Basically the same thing, but slightly different. So a source network, we'll be talking about mostly with the PET data, where we've got a set of regions that serve as the source of a single component of variance. Functional network, we're going to be talking about Fmri data and a component of time course, and a set of regions that source that component time course. And I'll talk a bit more about that as we get further along. A few words on independence and the pursuit of independence. Sorry, I'm mixing which way this is going. So when we're doing ICA in medical imaging, we have hypotheses about what might be causing differences in our abstinence, but when we're looking at our brain data, we have to do this a bit more blindly, and we have to look for non-random variance structures, right? So we want to find sources of variance that are maximally non-Gaussian, right? Random variance is going to be normally distributed, so we're looking for non-random variance. We're looking for something non-Gaussian. And we also want to minimize the mutual information across the components and sources, right? And different ICA algorithms are going to pursue this independence differently, and it's really complicated sometimes how they go about it, but it's also not necessarily important to understand how it finds it, and just perhaps more so what it is finding. There are some limitations and restrictions, right? ICA does not work on truly random data. If your data set is full of truly random noise, ICA might not be able to help you. There are some algorithms that allow for a random source sort of residual component, but that's not typically standard. The user has to decide how many sources or components to solve for. Right? You just have to say, I think there's 10 sources or 20 sources in my data. Give me those. At the moment, we're restricted to linear mixtures of component variance. Right? Some people are working on nonlinear ICA algorithms. Problem there is that when we start talking about nonlinear space, we lose the singularity of an optimal solution. Right? There's many optimal solutions to model the variance, and so we have to start constraining our algorithms. Whereas if we just stick to the linear mixtures, the algorithms are pretty good at just optimizing for a single solution. And of course, the true sources are not going to be known ever, but we can do our best to replicate and validate and test as much as we can. To review, right? We have some variance that we would like to explain. We're hoping it's not random variance, or at least we don't think it is. So we think it has some structure, and it represents some mixture of underlying variance sources. Right? ICA is going to separate out components of that variance, identify the sources of where that variance is coming from, and it's doing that by pursuing maximal independence. Right? It's going to output two sets of loadings. Right? It's going to give us how much component variance was found in each source, a source loading, and how much component explains the total variance for each measurement or subject. Right? A component loading. We can skip that. So let's talk about ICA of PET. The question is going to be, is the regional variance in our PET scans random, or might there be some underlying sources to explain the variance across our brains? We're going to use ICA to analyze the patterns of regional covariances across subjects to look for independent sources of variance. And as Anna Rose mentioned, I'm going to be walking through some PHNO data, which is a radio tracer that binds to dopamine D2 and D3 receptors in a pretty unique and interesting way. that we think lends itself to ICA pretty well. What do we know about D2 and D3? I've lost my... So D2 and D3 are highly expressed in subcortical and midbrain structures that are central to addictive processes, broadly implicated in impulsive and compulsive processing. Right, so we've got, if we just look at the brain on this side, we've got D2 receptors that are highly concentrated in the dorsal striatum, ventral striatum, and a bit in the ventral pallidum, whereas D3 is really concentrated in the midbrain, with a few more receptors in the pallidum and less so in the ventral striatum, not much in the dorsal striatum. Right, so this distinct and overlapping D2 and D3 distribution circuitry suggests that these may be independent sources of variance in our PHNO scans, and ICA can hopefully help us separate them apart. What we know about D2 and D3 in cocaine use disorder is that we have lower striatal D2 availability in cocaine use. These findings typically come from raclopride studies that have an equal D2 to D3 affinity, but they also do not detect differences in the D3-rich midbrain. Comparison, we have higher D3 availability in cocaine users, and these findings come from PHNO studies. And PHNO has about a 30 to 1 in vivo D3 to D2 affinity. But we're also, previously some studies don't necessarily detect differences in the D2-rich striatum in cocaine users. And what's making me laugh about that is that the regional binding of PHNO reflects local D2, D3 concentrations. So in, let's say, the D2-rich dorsal putamen, about 90% of our PHNO binding signal is coming from D2-related binding. Right, so if there's a difference in our groups in D2 binding in the dorsal putamen, we should be able to detect it with sufficient numbers, perhaps, in our cocaine users. And similarly with our signals in D3-rich regions, there's not much D2 there, but most of the signals coming from D3-related binding. We looked at a sample of 26 non-treatment seeking individual cocaine users and replicated some of the previous ROI methods looking at PHNO. And we did find that, in fact, that sample was sufficient to detect differences in PHNO binding in D2-rich related regions. And interpret as lower D2-related binding in cocaine users. And of course, we replicate the findings of greater D3-related binding in the D3-rich midbrain. The question becomes, though, what's happening in these regions that are mixed D2 and D3 signals, right? Can ICA separate out how much of this is D2 and how much of this is D3? And does that relative proportion differ between our patient samples and our healthy controls? Have a quick look at some of just the methodological processing details, right? We're taking parametric images, registered to standard space and processed in SPM12 with a little bit of smoothing. We're masking out voxels that have lower binding potential than 0.25, right? The idea, again, ICA is not good with random variance. And so we think that these voxels that had too little binding are actually going to be greater sources of random variance, and we don't want them confusing our ICA, so we eliminate those from the analysis. ICA analysis initially, we used what's called the minimum description length criteria, which just sort of analyzes your data set before you even get started and makes a prediction about how many sources you have, and suggested we might have 3 components in our PHNO data, which we then extracted using one of the algorithms called Infomax, using the group ICA for functional imaging toolbox. How does ICA parametric imaging work? Well, for our PET imaging, we're going to take our parametric PET brains for each subject and just transform that into a voxelized vector. Right, so all on the x-axis is voxel binding potential on the y. We're going to do that for every subject. We're going to dump that information into the ICA, and it's going to output our unmixing matrix, where we've got sources and components. And so the unmixing matrix gives us the sources and component loadings, those actual values, and we can turn our source vector, reshape it back into the size of a brain, and plot out our component subject loadings if we would like. Did some extra post-processing in this project. We scaled our source maps to estimated BP units. Okay? And so the way we do that is we take the source map loading, multiply it by the component loading for our total group, and we've recaptured or reconstructed binding potential variance, or variance in binding potential units. We can compare, and we also did that on the ROI level to calculate D2 and D3 related binding potentials, and to compare those to regional estimates of D2 and D3 binding from PHNO that are reported in displacement studies, right? So they've done PHNO scans where they block out all the D2 and see how much signal is left and say, hey, it looked like 90% of the butamen was D2 related, right? So we can just perform that analysis and get our ROI estimates of how much D2 or D3 was in our sample. You can look at our first source. We're calling it the striatal-palatal source network, and that source was located in the ventral striatum, sending out into the dorsal striatum. Sorry. And when we look at how much BP was explained by this source in each of the different regions, we found that it correlated very strongly with how much of the ROI estimated, the fractional displacement estimate of D2 was in each of those regions, right? Which allows us to say we think that this source is related to D2 binding. Found that there were lower component loadings in the cocaine users, right? Which would also then translate to having lower D2 related binding in our cocaine users, which is obviously consistent with the widely established literature. The second source, called the palato-nigral source, the source is smaller, but it came from midbrain regions, extending up a bit through the ventral striatum and into the globus pallidus. And again, looking at the contribution of BP variance related to ROI-based fractions of D3 related binding in those regions, we also again found a significant relationship, letting us make the assumption that this component and source component reflects D3 related binding. Found this component, there were higher component loadings in our cocaine users relative to healthy controls, again consistent with previous PHNO literature of greater D3 availability in individuals with a cocaine use disorder. Third component came a bit out like almost VTA-ish and up through the very ventral striatum. So it encompassed mostly D3 rich regions, but wasn't really correlated across all of the ROIs we looked at. It was also a relatively weak source of binding potential across our ROIs, and there was no group difference. We have since done other ICAs of our PHNO data and have just started getting 2 components out, and we very much just get those first 2 components. And it looks like this one gets added in with the D3 related source. I also found that our component loadings were associated with years of cocaine use in our sample, and specifically and differentially. So the D2 related source was negatively associated with cocaine use years, whereas loadings on the D3 related source were positively associated with cocaine use years. And that association with the chronicity was not found in our standard ROI based analysis of the ROI binding potential values. So it added a bit of findings. Oh, right. So what about binding in our mixed regions? So we reconstructed the ventral striatum and pallidum component binding potentials. So if we look back at just the raw binding potential in our ventral striatum, about equal between healthy controls and our cocaine users. But when we look at which component of that was D2 and D3 related, we saw the pattern of greater D3 related binding and lower D2 related binding in the striatum, though these didn't achieve significance. Whereas in the pallidum, we saw the same pattern and they achieved significant differences between our groups. Right? It allows us to say we think that there's greater D3 in the pallidum. It's something we're not allowed to say given the data we have from our normal ROI-based analysis. Also less D2. In summary, PHNO has a unique binding profile that lets us assess both D2 and D3 receptors. ICA blindly separated those D2 and D3 related sources of binding potential. It was more sensitive to chronicity than the standard total binding potential values from our regions of interest. And the ICA estimates did suggest it could detect these bidirectional cocaine-related alterations in D2 and D3 in our regions of mixed PHNO binding profiles. What about PET tracers that don't have this mixed binding profile radio tracerness? So we've started looking at different radio tracers. In one particular project, we're looking at the serotonin 1B system and comparing it to cocaine users relative to healthy controls and gambling disorders. And this data is very much in process, but we're seeing multiple sources of serotonin variability with a tendency towards lower component loadings in the cocaine group relative to both healthy controls and gamblers, and particularly in this one source that's in the ventral striatum and a bit more in the orbital frontal cortex, and the second source that's located in the midbrain and goes out to the very edge of the OFC. In progress, we're excited about this work and looking forward to getting to the bottom of it. A second line of work that I'm actually getting really interested in is looking at UCBJ, and this work's being done by Song Fang at the postdoc at the PET Center. And so UCBJ binds to synaptic vesicle proteins, and so it's a marker of synaptic density. And he's looking at sources of synaptic density variability and comparing it to activity in resting state networks. And preliminary analysis is showing that greater synaptic density in a striatal source of variance is related to activity in something like the salience network. Again, really interesting work. We do have some synaptic density data in cocaine users that Gustavo Angrita has at the Cocaine Research Clinic. And this is a direction I'm hoping to take those data very soon. So let's talk about functional brain networks. ICA of Fmri. So distinct functions may be distributed across distinct sets of regions, right? That's sort of how we think the brain works, right? We think function A might involve the ACC and the OFC, whereas function B might be going from the ACC to the ventral striatum, right? So if we see lower singular activity in individuals with an addiction, understanding which other regions that activity is connected to can help provide insight into which function it is that's being impaired. Right, and we can do this using temporal correlations in Fmri. We measure the signal in the cingulate while someone's doing function A, and we correlate it with the same signal during function A in the OFC. And if these things are correlated, we're saying that they're connected, and perhaps this circuit is what might be getting impaired in individuals with an addiction if this happens to be broken. Whereas compared to the signal in the ventral striatum, which may not be correlated during function A. The Fmri bold signal is a summation of local neural activity, right? What if we ask someone to do A and B simultaneously? What happens in the cingulate? Right? Bold in the AC would be a summation of those neurons. Right? In a 2 millimeter voxel, we're talking about hundreds of thousands of actual neurons in some parts of the brain. They all can't be doing the same thing at the same time. And so the bold signal we measure is just a linear combination, we think, of the different processes that are going through that voxel at any given time. Right? And it's not just two sources. And it can be really challenging to isolate discrete functions during an fMRI task as best as we can design tasks and design our contrasts for analysis. We can't really control for all the different sources that may be happening through every single voxel at every time. And so we can think of the fMRI bold signal as a mixture of all of these functional components that are going through the brain, right? And different mixtures at different places. Talk about functional components in a different way. An example I like to use is traffic. I don't know how familiar all of you are with the geography of Connecticut, but we're in New Haven, and so the question might be, where else in Connecticut might the traffic in New Haven be connected to, right? So we can measure traffic in Hartford, our other biggest city, and those two signals seem to be correlated, and we would say, yeah, sure, New Haven and Hartford traffic is connected, about an hour away. 45 minutes. We can also look in Greenwich, which is sort of on the way to New York, and say, yeah, there's some correlation, but it's a weaker connection. And we can also look at Ledyard, Connecticut, on the different side of the state, and say, New Haven traffic's not connected to Ledyard. And for those of you who don't know, Ledyard's where our casinos are located, so everyone goes there at night. We can also ask, what are the sources, right? What are the components of traffic in Connecticut? And we can put out those traffic signals through an ICA. And say, what are the different kinds of traffic that we see? And we can see that there's, small bumps that happen early in the morning, early in the afternoon, and they happen all over the state, a bit more in our highly populated areas. But then we can also see a greater signal sort of right around rush hour. It's super heavy in our populated areas. And then other sources, spread throughout the state. So we can test models or make inferences about the types of traffic we think each of these source components represents. Right? We can think this is school-related traffic. It happens everywhere. It doesn't happen that much, and it happens about the time all the school buses are on the road. Right? We've got our local commuters, and we can come up with explanations of what we think is explaining our other traffic components. Right? We're talking about functional networks and networks of function. Where we're not making, IC is not allowing us to make an assumption about connectivity or effectivity, right? A functional network is not a connectivity network. We're not saying Hartford school traffic is going to New Haven or affecting New Haven school traffic. We're just saying that there's school traffic happening in both places at the same time. So networks are types of cars, not cars traveling from place to place. And so a functional network is a network of sources of a component function. All the sort of the keywords that we use over and over. And so it's not that difficult to translate this to fMRI, but the basic idea being that the source of a functional brain network is a brain function, right? It's the process that the brain is working on that's causing the temporal change in both signal that is causing the variance that we're trying to measure and capture and structure. So what sort of functions do we get with our functional network? So this paper is getting on 10 years old, was a mass meta-analysis that took over 8,000 studies and looked at the peaked activation maps and ran them through an ICA and ran regressions with what tasks people were doing when they got those activation maps, and sort of ran a regression to say which brain networks were associated with functions, right? So if we look at, sorry, this is a bit blurry, this sort of block down here of interoception and emotion related networks, right? These are our top 4 networks here, right? Medial, frontal, salience, subcortical networks, right? And so we can go across the way, we can start to dissociate when we find this network in our data, look back and see what other people have found it might be up to when it's performing their tasks. What does the ICA of an fMRI data look like, right? So our input is our bold time series, and we transform that into a voxel by subject matrix, right? It's the same idea with PET, except now we've got subjects as well, right? These 30 or whatever subjects for every voxel. And we're going to do that for each time point, right? We get well, nowadays we get a scan every second. So this would be every second's worth of data. We enter that data, well, first run a PCA because that's a lot of data, and then run it through an ICA, and it just, again, is going to give us an unmixing matrix. Now we have to do some extra work to back reconstruct subject level data, right? But eventually we get a subject level source. Each person gets their own frontal parietal network, and each subject gets their own component time course of how their signals were changing over time through our scans. And we can just run our standard linear model analysis on those time courses, right? We can do a modeled HRF of what they were seeing or what they were doing in the scan at the time, run the regression, and if the component time course matches the modeled HRF, we can say that that network is related to whatever was happening at that time during the scan. Let's go through a quick little example. Right, we've got a response inhibition go-no-go data in young adult drinkers. Using a standard analysis, this group had reported widespread reductions in inhibitory activity that was related to the initiation of drinking. Right, we got lots of regions with lots of functional implications. So we took a similar data set of college freshmen or sophomores, I think, again with go-no-go data, and ran it through an ICA. And what we found was that it was the right frontal parietal network that was reduced on average, and again, sorry this is small and blurry, but was reduced on average in binge drinkers compared to those who were not binge drinking. But also it was reduced in response to committing errors in people who then escalated to binge drink or escalated in their binge drinking in the following year. And so we can ask ourselves, I mean, we all know what the frontal parietal network is these days, usually, but it's related to, just reasoning, inhibition, and attention, right? No surprise. But we couldn't get this information out of this standard analysis. But we also have other networks that were involved. We saw no differences in the default mode network, right? So it wasn't related to, let's say, binge drinkers just weren't the kind of people who pay attention to go-no-go tasks during the scan, right? Everyone's trying equally as hard. There's also no difference in a temporal occipital parietal network, right? Which is processing stimulus discrimination. The go-no-go we're using is X's and K's, which can be happening very fast and hard to tell apart. But there's no perceptual differences in our binge drinkers or those who escalated their binge drinking. So the ICA of MRI allows us to sort of parse apart these different components of a response inhibition or just go-no-go performance, right? Even a complex or even simple tasks. It helps us tease apart what might be happening. So our main interest, or one of my main interests, has been executive control alterations in substance use disorders, right? a lot of things go into executive control. They all seem to happen in the same parts of the brain. When you're running a task, it's really hard to isolate one of these from another experimentally, and it would be great if we could start teasing apart some of these effects to get a better understanding of what might be causing executive control problems in individuals with an addiction. So if we look at the now classic three-stage addiction model, we're really just talking about this one part of it, right? Impaired executive control. We think there's blunted top-down control, particularly in individuals with cocaine use disorder. What we think that would look like is lower engagement of our cortical control networks, right? Frontal parietal things, medial frontal, maybe the salience network should be blunted in our individuals with addiction, according to this model. But it might also just be a lower default mode suppression, and we'll talk about that more later. But the idea of just being a blunted global functioning might lead to blunted executive control. The other side of this model is that we have this greater bottom-up reactivity, which we just think will look like a higher subcortical network activation. The rest of these data, I'm going to be talking about Stroop task data, right? Just classic color word Stroop, no drug words. They're performing silently in the scanner, and we collect performance immediately before and after. Just some basics on the methods. Data is processed in SPM12. ICA again is performed in GIFT. And then we just run the temporal regressions to look at task relatedness to incongruent words for each of our components. At the beginning, right, the Stroop interference brain activity pattern looks like this in a standard general linear model. Right, these are 20 treatment-seeking individuals with cocaine use disorder. They entered a clinical trial that we were scanning at pre-treatment. We saw no differences in just average Stroop-related activity in this sample of cocaine users. And we also saw no difference in performance measures. Right, reaction times of error rates were the same. Got us wondering if maybe perhaps components of these processes might be different. So we ran that data through NICA. I found 5 networks that were related or engaged by performing the Stroop. We saw no differences in any of our executive control related networks between our cocaine users and healthy controls. And there's also no difference in the striatal network engagement. Looked like it was getting close, but didn't quite get there. So both of our hypotheses testing that model sort of turned out inconclusive, or to say the best. But what we did see was there was a differential relationship between functional networks and behavior. And that in the healthy controls, and we're going to be looking at, so down here we've got network engagement on the X, Stroop interference reaction times on the Y. So greater engagement in healthy controls across all these networks was associated with faster interference processing. An incongruent word, networks activate, they finish resolving the conflict faster. But in the cocaine users, greater engagement was associated with slowing down interference processing, particularly, or I guess just especially, in this medial frontal network and our subcortical network. Got us thinking that this greater activity in cocaine users was a reactive or an interruptive response, where in the healthy controls, greater activity was a resolution-based response, right? The activity was doing something different in our two groups. I think another part of that initial analysis, again, as I mentioned, these were treatment seekers. In the linear model, we found that greater Stroop-related activation in the right dorsal striatum was predicting abstinence during treatment. Got us wondering, what is that part of the striatum doing during the Stroop that is helping people in treatment? That part of the dorsal striatum was integrated into 3 of our networks. So we split the sample in half to those who responded and those who didn't. Ends are getting pretty small, but it's still an interesting analysis to run. And what we found was that the treatment responders showed a greater subcortical engagement compared to non-responders. And in fact, the non-responders weren't engaging their striatum network at all during the Stroop. This is counter to the hypothesis that the hyperactive subcortical network might be disrupting executive functions. It seems like a not active striatal network, at least during Stroop performance, was associated with not responding during treatment. And what we started thinking was that perhaps this bottom-up alert mechanism is what might be disrupted in cocaine use disorder. That's not impaired executive control, it's just that executive control never gets told that it needs to come into action. And that these greater alert signals in cocaine use slowed conflict responding, right? Perhaps to avoid errors and actually process the information, and that the healthy levels of alert signaling was associated with a better treatment outcome. And so we want to take a look at this from a different example, from a different angle, and use dynamic causal modeling, right? Just an input-output modeling of neural propagation, which allows us to understand how networks or components or regions may be affecting each other from region to region. Right, and we can look at just baseline activity, but what's more interesting is to look at context modulated effectivity, right? How do these networks change in response to the Stroop conflict? So this was a separate sample of non-treatment seeking cocaine users. And we extracted more ICA sources because as time has gone on, more seems to be better. And it separated out what was our sort of bilateral frontal parietal into left and right, and this bilateral IFG component. And we still, again, got our subcortical network. And again, we didn't see any difference in engagement in these components during Stroop performance. Some trends, but nothing significant. But when we look at connectivity, starting with our healthy controls, we're looking at sort of green arrows and red arrows. We can think of our subcortical network sending alert signal to the bilateral IFG sort of conflict resolution network, let's say, which then sends a signal over to the right frontal parietal, which then sort of distributes this inhibitory signal. So we're like, okay, we're processing this information, we're done. By comparison, in the cocaine group, the striatal network alerts both the left and the right frontal parietal network. The IFG signals head to the left frontal parietal instead of the right. And we have this strange pattern of the inhibitory signals of we've done processing this information. But I think this is a good point to stop and step back a bit and think again about what we're talking about, right? We're talking about source dynamics of Stroop control, right? While these networks are sourced to specific parts of the brain, what they represent is actual functions, right? They're source functions. When we think back, we have no differences in regional bold signals. We saw no performance differences between our two groups. We actually don't even see any differences in how our networks are engaged during performance. We saw some differences in relationships of how each network is associated with performance, but the performance itself didn't differ. But what seems to be different is how the component functions coordinate to resolve conflict, and it seems to be markedly different. Right? If we had to draw a picture of what we think inhibition looks like in the brain, this is sort of what it would look like. And something else entirely different is happening in cocaine use disorder. I mentioned the default mode network earlier as a different part of our hypotheses. We're doing okay. So the default mode is a core resting state functional network, and during task performance, default mode is suppressed, right? It becomes more negative. And how much it gets depressed is a marker of global executive function. And so if you've been paying attention through the data I've been showing, our cocaine users reliably tend to be lower in network engagement across networks, across studies, but it never achieves significance, right? And so we think maybe this default mode suppression might be better capturing a difference in global functioning between our cocaine users and our healthy controls. We also wanted to explore relationships with D2 and D3 related binding, right? We've got PHNO scans. And just explore the whole space of how these things might be related. Let's go through the outcomes. Again, this is a different sample of, again, non-treatment seeking cocaine users. And again, small n's, but still really interesting. No surprise, we replicated higher D3 related availability in the D3 rich midbrain. And again, in this sample size, we weren't able to detect a significant difference in D2 related binding, but it tended to be lower. And again, all of this is consistent in prior literature. In terms of default mode suppression, the cocaine users tended to exhibit generally less default mode suppression during the Stroop task. Didn't achieve significance. What did achieve significance, though, was a difference in suppression in response to an incongruent event. Our healthy controls turning off or suppressing their default mode to process an incongruent stimuli. Our healthy controls didn't really. Performance, no surprise. No difference, right? No difference in error rates, no difference in interference, delays in processing. So we want to look at the relationship between all of these variables. We start with default mode suppression and performance. For healthy controls, greater suppression of the default mode was associated with longer delays. We're looking at the gray line. We've got engagement of the default mode becoming positive, which is less suppression. being faster, or I guess more suppression being longer to be consistent. In the cocaine users, greater suppression tended to be associated with shorter delays. Not significant. And both of these findings are actually consistent with, we think back to that initial finding, the inverse of the executive control networks, where we saw greater activity associated with faster things. So default mode suppression is not related to error rates. Okay, so default mode suppression and D2, D3, starting with D2. In healthy controls, more D2 was associated with less suppression. So we've got D2 residuals. So these are residuals. This was all ran as a masked multivariate mixed linear model. So this would be D2 related influences controlling for any influences that might be associated with D3. In our healthy controls, more D2 was associated with less default mode suppression. Again, more negative being more suppression. Cocaine use subjects, no association between D2 and their default mode. D3 related binding, sort of the opposite. No association in the healthy controls. But in our cocaine users, greater D3 was associated with stronger suppression. So more negative default mode, more D3 in the cocaine users. And finally, D2, D3 in performance. Again, D2 in the healthy controls. Greater D2 was associated with shorter delays. Got D2 here, interference delays. Interference delays are getting shorter with D2 going up. And no association in the cocaine group. Again, error rate's not related to anything. And the opposite again in D3 related binding, where we see no association in the healthy controls, and in greater D3 being associated with shorter delays in the cocaine users. We're trying to think of a way to synthesize all of this information. Came up with a moderated mediation. So if we look first at the default mode, how the default mode is related to behavior and how D2 might be influencing that, what we find is that the default mode engagement has a significant indirect effect through D2 mechanisms on interference delays, but only in the healthy controls, right? Which is just what we've been saying for the past few slides, that everything in the healthy controls is D2 related. But this did differ from no indirect effects, in the cocaine group. So if we wanted to describe this effect, I think what we're saying is that D2 mechanisms facilitate faster conflict resolution with less suppression of the default mode. One thing about the D3 related moderated mediation, we saw default mode engagement had a significant indirect effect through D3 mechanisms on interference delays only in the cocaine group. Again, not surprising given what we've been saying. And this differed from no effect in the healthy controls. In summarizing this effect, I think it means something like that D3 related mechanisms are facilitating faster conflict resolution in the cocaine users, but with more default mode suppression. Right, so some very intriguing directions for further analysis. And that these two systems seem to be doing something very differently in these two populations. Do some summarizing. Addiction is complex, multifaceted disease, right? Substantial variability in individual disease profiles, motivations, and functional impairments, right? And improved understanding of what these many different sources of variability are and what they're related to and how much variance they can contribute surely would be helpful in a clinical setting. We might need to start rethinking the rewired addicted brain. Right, so how components of information are distributed across circuits may have changed. Right, school buses are taking people to casinos, possibly. The relationships between network activation and performance. And how information flows from circuit to circuit may have changed. Right, people are dropping their kids off at school on the way home from work. Everything's out of order and all mixed up. It'd be nice to understand the sources of addiction toward prevention and treatment. Right, are there at-risk source profiles? Are there certain sources that dominate in youth who go on to develop a substance use disorder? And something I'm really interested in is looking at the source dynamics and how they might change during early abstinence towards a sustained recovery, right in those early initial weeks of the brain undergoing various chemical changes. What might be happening to the source dynamics, and how can we improve those towards sustaining recovery? And understanding the sources of addiction function may more easily translate to a clinical application, right? Instead of saying, well, your striatum's too active, we need to fix that. Sort of like you're processing conflict out of order or something. At least from the way I think about it, and I'm not a clinician, I feel like that might be more helpful. A few final comments on ICA in general. Not all variance is created randomly. Some is. But underlying sources of variance suggest that we either have imperfect or imprecise measurements, right? We need to be looking at smaller and smaller pieces of things. total scores can be helpful, but subscales provide a greater precision. The most variance explained is not always best, and this is sort of one of the questions I always get, right? So analysis fitting the dominant variance vector or distribution, something like principal component analysis, might actually still be mixing true sources, and it actually probably usually is. All right, and so mixed source profiles can help if what your goal is just to explain the variance for most people, or explain most of the variance in your data. But from my perspective, understanding how those underlying sources are mixing in most of the people leverages all the variance in your data set. And I like to say that many independent small effects can contain a lot of information that adds up to a big effect. And a few things about ICA. Data-driven and blind does not mean hypothesis-free. Right? ICA is best applied when we have a rationale for why we think there are sources and an expectation of what types of sources we think we have. And sources are still going to require replication, validation, and even then still other support for your interpretation of what they mean and where they're coming from. And so with that, I'd like to acknowledge all those who mentored and helped me along the way and contributed to that breadth of work. Robert Mallison, Rich Carson, Mark Potenza, and Vince Calhoun, as well as many other individuals, including those in the Impulsivity lab at Yale, the Cocaine Research Clinic, Magnetic Resonance Center, and the Yale PET Center. Thanks.
Thank you very much, Patrick. Maybe some people can unmute so you can hear the clapping.
Thanks.
And I'll open the floor for people to ask individual questions. I always have a couple, but please, other people go ahead.
Patrick, great talk. This is Paul.
Hey.
It's a little off topic, but it's about the variance that we see, that I tend to see in more vulnerable, quote unquote, vulnerable populations. So like more Q reactive or more people with like extreme stress in their past. But it does seem to be that those groups have more variance overall. You might see a cluster around the mean, but then just tends to have more standard deviation or standard error of the mean, whereas like the less vulnerable group, and maybe you see this in the healthy controls as well, tend to be more tightly clustered, less variance. So when thinking about these, ICA analyses, like, do you try and take that into account? You have these subgroups within your cocaine use disordered populations. So it's not just as a whole, the cocaine use disorder population, but the groups within. And if I totally missed it and you do account for all of that, let me know.
No. I mean, I think that's a great question. I think people have been working on just that sort of problem, right? So for me, the answer is, it's not that there's more variance, that population is more volatile, and the variance is more volatile, and the sources maybe more or bigger or something. But there are new methods coming out. I'm forgetting where the paper was published or wrote it, where they'll do a cluster analysis first before dumping people into an ICA to sort of separate out different populations before finding the sources for those populations. So that could be one way of going about it, right? To separate out these extra volatile people and try to run an ICA on them separately and sort of not mix them in with everyone else. But yeah, I mean, I think it's the nature of the beast that the amount of variance in each person is not going to be the same. And it's going to be wildly different depending on the population of how much variance in variance you can have. Does that make sense?
Thank you.
Patrick, it's Anna Rose. Have you been fiddling yet with trying to push these networks around with pharmacologic interventions?
Not personally. No, it's certainly something. It is something we're very much interested in. I think some of the newer systems we're looking at, so some of the like you mentioned, my K and the mGluR5 system. So there's been some work with ketamine and looking at the effects of ketamine on mGluR5. And I think it'd be super interesting to look at, how those networks might be changing on something like that.
One of the things that's interesting, and we talk about it from time to time, is whether the face of recovery in these networks, will be a return to something that looks like healthy controls, or whether it's going to be a different looking creature. I'm sort of reminded of the work with Hugh Garavan with the cocaine patients who were in a long abstinent group, sort of a self-selected group, absolutely. But they seem to have, if anything, enhanced recruitment of inhibitory regions. And it's a chicken egg question for if that's how they came to be longer absent or what, but what are your thoughts about, from what you've already been doing, because you've been looking for this early absence, later absence, and maybe prolonged. And I know it's always hard to find a group of prolonged abstinent patients to be able to see what the endpoints will be. But do you have thoughts about that, about whether you're going to be trying to look for a recovery that looks a lot like the healthy controls, or will it be something that actually will be a different creature?
Yeah, I think that's one of the ultimate questions of how do we get people recovered on a long-term basis? Where do we have to push their brains to? I guess to piggyback on that last question. I think there's been some recent analyses that are also consistent with Hugh's earlier findings that a recovered brain is hyper good at being healthy, I guess is one way of putting it. But I'm stuck in the world of variance. I don't think there's one answer for everyone.
Yeah.
Right? I think it's going to be tough.
We embrace heterogeneity. I think it's one of the things that actually not only will be informative, but will be really important as we do go toward interventions, to understand that we really do have a lot of people who are very different under the same crude umbrellas of a diagnosis. And, it's where you start, but basically it is a complexity that we have to deal with, whether we're talking about a behavioral or a brain intervention with a medication. So that was one of my curiosities.
No, it's a really good question that I think everyone's trying to to get to the bottom to, so we can help a lot of people.
And the other thing is just an individual curiosity that we have, when the default mode network is there, and sort of in some of our paradigms where we're asking people to attempt to inhibit their brain response to things that remind them of cocaine, for example, we can see huge activations in lots of places, actually, and including some of the places that are quote unquote the inhibitory circuits. But the interesting thing is, or the challenging thing is, once you look at outcomes, you can see that for some people, those activations and those networks are actually sort of a signal of struggle or futility. In other words, there's a lot of activation going on and a lot of crosstalk going on, but in fact, it's associated with people who have poor outcome. Conversely, sometimes people who have a sort of a lower, quieter, potentially more efficient brain response have a better outcome, and that opens the door for all kinds of annoyances, like, if something's not the thing that's predicting, then it's the opposite, and it gets to be a little bit wild. Do you have any personal philosophical thoughts about the posterior cingulate?
I think a lot of connections go through the posterior cingulate. But I'd also say, what was I going to say? But yeah, I think...
Or just about DMN. What if you would like to comment on that as well?
Well, I guess, I started using ICA hoping to find answers, right? And I think more often times than not, we're finding more questions of how come, it's a struggle response is what's being shown in this particular circumstance, in this particular population. And, I thought the opposite was going to happen, right? We didn't, other than that one default mode difference on incongruent responses, we had no differences between our functional networks and this group in our subjects, right? It was completely the opposite of what we would have thought.
Yeah.
So just more questions of trying to understand the nature of these things now, where we thought they might be answers. They're more questions.
Right.
Before we run out of time, I wanted to get Patrick's reaction to a very provocative commentary by George Koob in the most recent Biological Psychiatry. And he's focused on the rat model of exactly what you've been, or close to anyway, what you've been studying, and it's a proposal for a microcircuit in the basolateral amygdala. And I was just wondering what are the size constraints that make contrasts and comparisons between the heavily studied rat limbic system and the potential for a curving engram, and what you can reasonably approach in human beings. Could you comment on that?
I haven't seen that specific article, but I think, I usually like to approach these questions starting with preclinical translational research and sort of using that to sort of inform what we think we might find. And recognizing that there are going to be limitations in terms of, the vasculature is even different, let alone the neural circuitry. And so approaching, those questions and those data from preclinical research with full awareness of the limitations of how much we can actually translate it, I think is a healthy perspective to have. But that doesn't mean it can't be helpful at all, let's say, right? We've learned a lot from animal models, and I don't think we need to disregard them.
Is there hope that as the technology improves, that we'll be able to make more comparisons between the animal models with the possibility of biochemical analysis and the human research that it's very hard to make a comparison, but is that kind of a long-term goal?
I mean, I think animal researchers, for the most part, are doing their best to model human behaviors in an animal model. They appreciate that what they're doing is try to emulate and provide information that might be helpful in humans. And so to the effect that the technology from their end, they're always working on genetic knockouts and optogenetics and different things to try to get a better model of human behavior. And I think they're making advances in good directions. That helps.
Hi, Patrick. This is Bob Mack. Couple questions for you. Really excellent work. I really enjoyed your presentation. I think my first question is, in your being able to segment out the D2 and D3 with PHNL, it looks like the change in D2 that you're seeing is less than what people have reported with raclopride. So, I mean, how much of error is introduced by having to separate out a D2 signal with PHNL?
That's a great question. I want to rely on my ignorance of the kinetic differences between the two tracers and why perhaps PHNO binding to D2 might provide a different amount of difference in cocaine subjects, just in terms of the magnitude of the measurement. Is kind of what I want to default to because I don't know better. But I think that's a very valid point and a good question to make. I mean, I would say, if we don't separate out the D2 signal, we don't see any difference. So we're at least being able to recover some of that signal from a PHNO scan using the ICA to identify it.
But you're likely underestimating the true reduction in D2 with PHNO, right?
Yeah.
Yeah.
Well, yeah. I mean, and just from the normal linear or GLM ROI based model of D2 related binding in the dorsal striatum.
And the other question is, these were patients who were not seeking treatment, right?
It was a little mixed. Yeah, some of the earlier data was treatment seeking. We were recruiting people going into clinical trials at the Psychotherapy Development Center. But we've replicated much of it in people who are not treatment seeking. And a lot of my work these days is in people who aren't treatment seeking.
Right. So
yeah, I think it's an important distinction to keep in mind.
Yeah. So if you have a group who say they are in abstinence and prolonged abstinence, where you're likely to see a change in the D2 population and a change in D3 population, but they're not likely to change at the same rate. Do you think you will be able to tease out that difference with PHNO?
Would like to think so.
Yeah,
that's fair. I think that's a really interesting question. And again, what's one of my interests is looking at how these, neurochemical and functional systems are recovering over time. And I think that would be a fascinating experiment to run. To see
my group has been focused on making selective ligands for both the D2 and the D3, and I think, with respect to the data you showed, the direction, I agree, they're in the right direction. The D2 is down, the D3 is up. I think you may be underestimating at least the D2 and maybe the D3. I agree with what you're seeing, but the numbers may be a little off. And I think if you had selective ligands, you could probably get a better measurement of the D2 and
yeah. I think another important point to just make is we're doing all of this at the whole brain level. Right, so we've got a lot of smoothing and modeling going on, and I think that wipes out a lot of our effect sizes compared to normal, time activity ROI modeling. So that may be contributing to some of the difference in effect sizes, but I can appreciate that.
It's fantastic work. I really enjoyed it.
And it gives us a great excuse to have you come back again, Patrick.
Thanks.
To give us a follow-up.
Would love to. Absolutely.
Any other questions from folks? We're just a little bit after 4, but I don't want to cut anyone off. We have a couple of minutes if somebody has a burning question.
I was just going to follow up on Bob. This is Rich. And Bob, we are looking forward to your selective D3 and to see if you can do in chemistry what Patrick has done with ICA.
Woohoo!
Take those apart. And that will obviously...
I'm looking forward to it, too.
I know you do. The other subtlety is the agonist versus antagonist nature, and I think that opens up a fabulous one. And, we've long since looked to be able to do depletion studies and things like that, which we haven't been able to get funded. But to differentiate the effects, what we'd see with raclopride versus PHNO, be able to take that all apart. So it will be fun. It will be better with a selective D3 tracer. Absolutely.
Patrick, what you should have said in your answer is, talk is cheap, Bob. Why don't you hurry up and deliver the D3? Stop talking about it.
Or you could just say, Bob, I'd love to be one of the first to test your tracer. There you go. All right. Well, this has been really fun and a highly creative use of ICA in both worlds. Renaissance man. Thank you, Patrick. We appreciate it.
Thank you so much for having me.